quite some time ago, but in the first lab in Netherlands. And what we did at that point, this was the first lab, is to produce those structures that we call blastoids from mouse stem cells. And they self-organize quite well. You can see that it's very recognizable morphologically. You've got the 3 cell types that form, and because it's pre-implantation, we can actually transfer them into the uterus where they implant. And those blastoids are quite unique in 2 respects. First, they are complete. And second, they represent the pre-implantation stage, so we can actually understand this process of implantation into the uterus. And of course, what we did is to crack open those uteri a couple of days later, and the structure is growing, differentiating. There's some morphogenesis, but the process is quite disorganized. And at this moment, nobody has formed a mouse out of a mouse blastoid, although a lot of people are trying. And we are still pursuing to understand how what is missing in this minimal system in order to make sure that one day we actually make a mouse out of it. And we have a lot of progress on this. We're actually not very far from getting things right, but there's still a bit of work in front of us. Nevertheless, by using this system, we actually managed to understand some important mechanisms, and this is what we focused on in the mouse system for the last 10 years. And we understood a couple of things, that especially that those cells that are here, that are going to form the fetus, are very strongly acting to regulate the development of the trophectoderm. And there's a couple of signalings that are coming from the inner part and that are regulating the proliferation and the self-renewal of those cells that are here, that are CDX2 and KLF6 positive. And of course, if you regulate proliferation and self-renewal, this is to maintain the stem cells that are going to form the placenta. And this allows for the growth of the organ. The second thing that we found out is that those signals that are coming from the epiblast are regulating epithelial morphogenesis, namely the formation of this cystic structure here. And this allows to pump water and to form the cavity. And very interestingly, when you pump the cavity, you spontaneously distance a little bit those cells from the signaling center. And by doing so, those cells are slightly differentiating, and this allows for patterning. And this is the first patterning that happens in the embryo. And those cells actually become sticky, and these are the cells that are attaching to the uterus. So this is the second phenomena that we came across. And the third one is that there's also a trophic effect. By maintaining CDX2, which is a transcription factor, the epiblast here is regulating indirectly the production of some factors, including Wnt, that are directly acting on the uterus. So this part is sticking in the mouse, and this part is instructing the uterus to form the cocoon around it. It's a very clever system, and that is actually extremely simple, but very interesting. And overall, what we understand is that at the very beginning of embryogenesis in the mammalian systems, the goal of the embryo is actually to form those extra-amniotic structures that are going to, as an investment in order to create this interface with the mother, and of course, the formation of this placenta is giving back a lot in the next few days and months by allowing for the fetus to grow. But the first investment that the embryo is making is to push the development of those extra-amniotic structures that are going to enable the formation of the placental-maternal interface. So this is for mouse, and of course, many processes are developmentally conserved between species and between mouse and human, and a lot of the signalings that I showed you before are actually present also in the human blastocyst. However, during evolution, there are a couple of very interesting things that happened, and I'd like to highlight 2 of them. The first one is the direction and the depth of interaction. I told you that in the mouse, the sticky cells are formed on this side, and you can very nicely see this is the lining of the uterus, and you can see that the blastocyst is attaching via the sticky cells that are on the opposite side of the epiblast. And in the human, it's actually the opposite. So during evolution, the whole system flipped around by 180 degrees, which is an extremely drastic event, and the embryos started to stick from the other side. Actually, this is more or less the canonical path. This is rather an exception in the animal kingdom. So, of course, because of this, it's complicated to try to understand humans with mouse. And the second thing is that I told you is that there's, in the case of great apes, we are very deeply embedding into the endometrium. And this is something that we cannot model with mice that barely invade. And we cannot model with many other species since this is actually great ape specific. So this led us to develop the system in the human. And it turned out to be like a very exciting moment in the lab because it turned out to happen extremely fast. We started working on this in about 2019, and by 2021 everything was finished and published, which is quite exceptional, and I hope it happens again in the lab one day. But the reason why it happened so fast is that we were standing on the shoulders of a couple of labs, especially the lab of Austin Smith in Cambridge, that had captured human embryonic stem cells in a so-called naive state that reflects on a very early stage of development. And by putting those naive cells in culture and using the knowledge that we had developed with the mouse, we could actually see that those structures are capable of self-organizing very closely into something that resembled the human blastocyst. The system is surprisingly efficient, and we are using a triple inhibition of the ERK pathway, the TGF-beta pathway, and the Hippo pathway. And the Hippo pathway turned out to be absolutely critical, and we are using a GPCR ligand called LPA in order to trigger the process. And then when you do this correctly, the cells actually remember where they come from, and they start to self-organizing very efficiently, about 80 to 90% with embryonic stem cells, but also with iPSCs, which are induced pluripotent stem cells that can be harvested from the skin of everyone. The structure is remarkably similar morphologically, and when you look at the three cell types, they are here. We have the trophectoderm here, the epiblast, and the primitive endoderm. There's a slight shift in the ratio of the epiblast and the primitive endoderm, and we are actually fixing this at the moment. This is already fixed, in fact. Yeah. But the first thing that we wanted to know when we actually saw this happening in the lab is to know if we actually formed the right cells. And there's been quite a debate in the field, but of course, single-cell sequencing is extremely important for this. And you can see here on this UMAP, these are the naive cells to start with, and they are fitting here. And within 24 to 60 hours, those naive cells are forming two cell types, one that is transcriptionally very close to the naive cells, and this is the epiblast of the blastoid, and one that is a bit further, and these are the trophoblasts. And in the second step, between 60 and 96 hours, those two cell types have a transcriptome that progresses, and there's a third cell type that appears, and this is the primitive endoderm. And this was very interesting because this is exactly the sequence that the blastocyst is taking. So the sequence is correct, and the timing is 96 hours, which is quite close to what we see in the IVF clinics. It's a little bit too long. It takes about 4 days instead of 3.5 days. But overall, the timing seems, is quite close to the blastocyst. So because we have the sequence and because you have the timing, we thought, okay, maybe we are not so far away from getting the right cells. But in order to really understand whether those cells are the correct one, first thing that you do is to spike some controls. These are post-implantation epiblast cells, post-implantation trophoblast cells. You can see that the transcriptome is actually very different. But of course, the right way of checking this is to project the cells onto a reference map. And we did our own reference map, but as soon as we published, there's a consortium that actually gathered that reanalyzed the data. And this was led by Frederic Lanner and Sophie Petropoulos, and it was published this year by Chen Zhao. So what they did is to compile all the single-cell sequencing data from the human blastocyst and some gastrula that were collected, and to look at the lineages formation. And you can see here first lineage specification with the trophectoderm that goes on the right side and the inner cells that go on the left, and then second lineage specification with the epiblast going on the left side and developing into the mesoderm, etc. And on the right side, the hypoblast that is going to form the yolk sac. And this happens between, so the first lineage is on day 5, first lineage specification on day 5, and second lineage on day 6 to finally get a blastocyst, a full-fledged blastocyst on day 7. And what you can see, or what they saw, is that the cells are very nicely nested in the right spots with the day 6 epiblast or day 7 epiblast here, the hypoblast here, and the trophectoderm here. They measured that 97% of the cells are actually have a transcriptome that is very similar to the human embryo. So that's a very happy moment for us. People actually validating our findings. There's a bit of an overfit here because those cells are quite different from those ones. So if you put some cells like ours, they naturally cluster with the closest ones, which are those ones. So it doesn't mean that the cells are the same, but they're actually very similar. So these are all interesting molecular and morphological features, but of course what we would like to know is if the structures are functional. And of course, as you can imagine, it is ethically unacceptable to transfer those structures into a human, and it is actually now forbidden by the International Society for Stem Cell Research to transfer them in any living uterus of animals or humans. And I participated to the establishment of those guidelines. But what we can do is to grow some endometrial cells as organoids. This is work that has been done by Margarita Turco and Hugo von Kelkom. And this is in the style of Hans Clevers in Matrigel with a couple of molecules, R-spondin, etc. And you can massively expand those biopsies, and then you can plate the cells in order to facilitate the depositions of the blastoids. And what we observed is that the blastoids are incapable of attaching to those endometrial cells unless they have been previously stimulated with the hormones of pregnancy. Progesterone, estrogen, and also a Wnt inhibitor. Happy to talk about the Wnt inhibition here. In that case, we have about 30% of the blastoids that attach and that start a process of invasion that is similar to the initiation of placentation. So this is in 2D, and now we are developing the system in 3D. And what was very interesting is that, as you can see here, literally all the blastoids are attaching via the polar trophectoderm. So we have a very specific process here on the endometrial side, where the cells must be hormonally stimulated to become competent for attaching the blastoids. And we have a very specific process on the blastoid side, where the sticky cells are only forming on one side, which is the side by which the blastocysts normally attach. So here we have a system that allows us to do a little bit of interesting science. Of course, from this derives directly the hypothesis that there must be, just like in the mouse, there must be a signal coming from the epiblast that, in the case of the mouse, maintains the stem cells, but in the case of the human, not only maintains the stem cells, but also makes some of the cells sticky. And you can actually visualize those sticky cells by a staining for NR2F2. This is an antibody that was, a marker that was found by Laurent David in Nantes. And you can see that those cells become NR2F2 positive and CDX2 negative. And so, of course, to test this hypothesis, the best way to understand what are the factors coming from the epiblast is to actually try to remove those epiblasts and see if something happens. And we did this in 2 ways. These are chemical ways. We also did some genetic ways. By inhibiting the STAT, so we just add one STAT inhibitor, it's called SC144, during the blastoid formation, or we inhibit the Hippo pathway by using XMU-MP01. And in both cases, by just adding one molecule during blastoid formation, the center cells do not form, and we have a large 95% of the cells are actually trophoblast. So these are empty blastoids called trophospheres, and when you deposit them on hormonally stimulated endometrial cells, they are incapable of attaching. So this very fits with the idea that those inner cells are sending some signals, and I'm actually happy to discuss what those signals are, because we have found at least a couple of them. We also added a couple of controls. One here, these are the trophoblast stem cells, the post-implantation trophoblast stem cells that were on the UMAP before. And we can form small aggregates also, and those post-implantation stage cells are also incapable of attaching. That's very interesting, because it shows us that there's a signal coming from the inner part that makes the cells sticky at a very specific time, and when those sticky cells keep on progressing, and that after they become incompetent for attaching to the uterus. So the embryo is creating a very short time window during which it has the capacity of interacting with the uterus and of entering into it. Yeah. So of course, this system is interesting, because now we can go into the genetics and find out what are the elements that contributed to the evolution of the specific mode of implantation of humans, and how this might have brought some advantages, but also some trade-offs, such as fragility. This is work that we are currently finishing, in which we were looking for the specific regulators of the polar trophectoderm attachment and invasion that led to the great ape-specific phenomena of deep embedding. So I showed you that there is like a window of implantation, and indeed, when you take very early blastoids, when 60 hours, some of them are implanting, but mostly not. And when you wait a little bit longer, you have like very nice levels of attachment to the endometrium. So this tells us that, you know, like the transcriptome is actually like evolving, and what we can do is to model this gene regulatory network using a single-cell RNA-seq and ATAC-seq. And I don't have to explain too much, but I guess, with the ATAC-seq we actually get the accessible sites of the genome. In those accessible peaks, we actually can find some DNA motifs, such as GCM1 here. And then by looking upstream, downstream, typically 15 kbs, we can actually like find genes. And by doing this step by step at the transcriptome level, we can actually create networks. And this is work that has been done especially by the lab of Steiner Aerts in Belgium and Barbara Treutlen in Switzerland, with some softwares that are now called Scenic Plus and Pendo. And by using those softwares, we can actually like generate those networks, and you can see very nicely the network of the trophoblast on the left and the network of the epiblast on the right. For the sake of simplicity, we removed the third cell type here. And you can see that between 60 hours and 96 hours, this gene regulator network is actually shifting to actually like upregulate some genes that are very central to the polar trophectoderm, such as GCM1, for example. And now, if we only isolate the trophoblast, we can look at it more closely, and we can actually see here GCM1 again, and you can see that this is, if you look into the embryo, not the blastoids, you can see that GCM1 is very nicely expressed on the polar side, and if you look on the other side, for example here, you can see TCF7L1, and you can see that TCF7L1 is largely more expressed on the mural side, so the opposite side. So this network is most likely a mural network, the opposite side of the sticky cells, and this network is most likely relevant to the polar trophectoderm. Now what we can do is to rank the transcription factors that are in this network by looking at the expression levels of the transcription factor itself, and by the expression level of the genes that they are putatively binding to. So GRHL1 is binding to 238 putative targets, and together they have a very high enrichment score, and then we can rank them, and you can see that the genes that are important for the polar trophectoderm are actually very different from the mural trophectoderm, and these are most likely the ones that are mediating this attachment and invasion. And what we can do next is to do such a cluster matrix where we cluster the transcription factors according to their common targets. So TFCP2 has 159 putative targets that are very much in common with GRHL1, which has 234, so it clusters together. And then when you do this, you can see that there are two different programs, one at the top side and one at the bottom side, and that we have all the genes here that are mediating the process. And now, of course, we can validate first a couple of things. So you can see that I'm a bit obsessed with GCM1 lately. So this is GCM1 here. And you can actually do the staining in the blastocyst. You can see that GCM1 is very nicely expressed on in the polar trophectoderm. This is data that was generated by Eva Monard and Laurent David in Nantes. And this is a late stage blastoid, and you can see very nicely GCM1 being expressed in the polar trophectoderm. So now we got interested into what those genes, what those sets of genes are actually doing. And in order to look at this, we went back to the embryo, the blastocyst, and we look at all the genes that are expressed on the sticky side and all the genes that are expressed on the other side. And we did some ontologies. And very interestingly, you can see that on the polar side, the genes that are specific to this part are regulating cell migration, cell adhesion, GTPase, Wnt signaling. I'm happy to talk about this again if necessary. Epithelial cell migration, EMT, placental development. On the other side, the cells are actually doing something else, TOR signaling, Hippo signaling, etc. So now it's interesting because we have like some gene sets of the embryo itself that are relative to the functions that we are looking for. So now what we can do is to plot those gene sets that are coming from the embryo onto our matrix that comes from the blastoid, and look how many of those 230 genes are actually like regulating cell migration or cell adhesion. And you can look at some enrichment scores here, and very beautifully you can see that those transcription factors are feeding into the polar trophectoderm gene sets of the human embryo, whereas those transcription factors are feeding into the gene sets of the other side. So now we know which transcription factors are going for which programs, and of course this informs us about doing the genetics. And so now we actually did, I think, like 9 knockouts, and out of those 9 knockouts, we found 3 that are extremely functional. So these are the ones. GRHL1, GCM1, and TCF7L2. 3 transcription factors that are very highly ranked, feed into those programs of cell adhesion, cell migration, and that when we test them into the implantation assay, you can see no hormonal stimulation, the blastoids do not attach. Hormonal stimulation, they start attaching, and then GRHL1 knockout go back to basal level. So this gene, this transcription factor that is expressed on the polar side is absolutely necessary for the attachment of blastoids to those hormonally stimulated endometrial cells. The second thing that we can do is to let the blastoids to develop just a little bit, 4 to 6 days, and to see how the trophoblasts are coming out from those blastoids. And you can see here that in the wild type case, the blastoids are forming those trophoblasts that are characterized by a downregulation of GATA2 and an upregulation of FCGB. SOX2 is an epiblast marker, you can forget it for now. So HCGB, this is of course like the hormone of pregnancy, the one that we detect in the clinics, and so these are the trophoblasts that are producing the cells, the hormones that we detect. So this is actually the pregnancy test is an implantation test. And very interestingly, when you look at what GCM1 blastoids are doing, you can see that they do not downregulate GATA2, and they do not upregulate HCGB, so they have a massive differentiation defect. Those cells are here, they are proliferating very nicely, actually very highly, but they are incapable of moving forward. GRHL1, very interesting phenotypes. GATA2 is going down, but HCG is not going up. So there's like less drastic, but still very prominent implantation defect, and those cells, those blastoids, as I showed you before, are incapable of attaching to the hormonally stimulated endometrial cells. So we have 2 phenotypes there that are clearly due to a lack of differentiation, And the surprise came from the last transcription factor, because this one is actually inducing a massive differentiation of the trophoblasts. The trophoblasts are just, if you knock out these transcription factors, they are unleashing, and they just differentiate much faster. So this fits very nicely with the idea that within this gene regulatory network, some transcription factors are pulling down, are here to allow for the GRN to progress, while at least one other transcription factor is here to slow down the progression of this GRN. And we think that this is by having those two counter forces of accelerating and slowing down the progression of the GRN that the embryo has the opportunity to create this window of implantation. So that's, of course, very interesting, because from the evolutionary perspective, you can imagine now that by having those two opposing forces, the embryos can play with it. So that they can extend it, they can shift it in time, etc., and do all kind of things that that can be very interesting for them. I mean, I shouldn't say that there's no intention here, but of course they have it brings the capacity of evolving. So the way that we understand it is that there are two different forces. TCF-cell2 is definitely putting a brake on it. TCM1, GRHL1 is accelerating it, and together they are timing the adhesion of an invasion of the human embryo. So in order to enter more into the question, we can now look at different species. Those different species are embedding inside the uterus at different times and to different depths and in different directions. Great apes, I told you, they implant very deeply, and the blastocyst attaches around day 7 to 11. We don't really know, and there's a lot of variation. Old world monkeys that are on this continent, especially the Cynomolgus macaque, it implants around the same time, but the depth is much more shallow. I'm happy to talk about a very interesting process. We actually invade the uterus via the stroma, and they invade the uterus via the endovascular system. And well, it's not black and white, but it's definitely a lot more for them on the endovascular one. And this is probably the stromal route. This strong affinity for the stroma is probably what allowed us to go very, very deep. And I'm happy to discuss why do we go deep. The physiologists actually know. And so these are the old world monkeys. And now the new world monkeys that cross the Atlantic actually attach but never enter the endometrium. The critical example is the marmoset. It attaches actually a bit later, day 11 and 12, and it never enters. So the blastocyst and the fetus is actually growing inside the uterine cavity, but not inside the uterine tissue. And of course, mice, as I told you, they are doing some weird stuff. They're actually they flip the axis and implant via the other side. So now we can look at the 3 transcription factors that we found to be central and essential for the implantation of blastoids into hormonally stimulated endometrial cells, and look at their expression in those species. So you can see these are the transcription factors that we found important for human, and these are the ones that are actually functional in our hands. When you look at the macaque that implants in the same way but doesn't go as deep, there's one transcription factor that is clearly missing, clearly lower, GRHL1, and if you look down, there are a couple of other ones that are clearly missing. If you look at the marmoset, other side of the Atlantic, 2 out of 3 of those transcription factors are actually not expressed in the blastocyst. And if you look at the other ones, there are also quite a few that are missing. And if you look at the mice, very interestingly, you see that in the polar trophectoderm, none of them is expressed, but if you look at the other side of the blastocyst, there are 2 that are very nicely expressed, via GRHL1, TCF7L2. So this is classical evolution. This is an heterotopy, translocation in space of the expression of a certain feature, in that case a transcription factor, that in mice somehow got to the other side, and then the whole thing flipped around. That's the way we understand it. Yeah. But very interestingly, there's one here, the one I'm obsessed with, GCM1, that is not expressed. We looked into the placenta into the blastocyst, you can see, so RE3 very nicely expressed in mouse in the sticky side, GRHN1 very nicely expressed in the sticky side, GATA2, GCM1 not expressed at all. And if you wait for a bit longer, in the placenta, you can see GCM1 is massively expressed. So here it's not an heterotopy, it's actually an heterochrony, where this specific feature has actually been shifted in time, not in space, and started to be expressed much later. So we went back to this GCM1 that is heterochronic and heterotopic in mice, and looked at it in those monkeys. So these are the monkeys that are on the other side of the Atlantic that attach but don't invade. And you can see that at the blastocyst stage, this is the trophectoderm here, GCM1 is absolutely not expressed. It's actually a little bit expressed in the epiblast that is going to form the fetus. But in the trophectoderm and in the sticky cells, we don't see anything. However, if you wait long enough and look at the post-implantation trophoblast, bam, GCM1 is actually expressed later. And this is like after implantation. So again, similar to the mice, those all the New World monkeys actually have an heterochrony as compared to the humans, that they are expressing the transcription factor later. It's not only GCM1. If you look at all the genes that are important for the human, they are not expressed in the blastocyst, they are expressed much later. So there seems to be a massive heterochrony here. So of course it leads directly to the hypothesis that during development, we know that those transcription factors are regulated by cis-regulatory elements, meaning genetic sequences that are enhancers, silencers, and so on. And we know that those cis-regulatory elements are the ones that are the main drivers of evolution. We know this from Drosophila for like 75 years. So our hypothesis here is that there are species-specific differences in enhancer activity for those 3 transcription factors that we have found, and that potentially played a role into the evolution of timing and depth of human implantation. So can we observe, the question is, can we observe differences in those enhancers? And the way to answer the problem is was actually to develop some deep learning models of enhancer accessibility. And the way that we are doing this is that we use the ATAC-seq data that we got from human blastoids. We built a model, and this is actually done along with Alex Stark at IMP. And the model that we trained by deep learning, so we do not understand what happens, but there's a model that comes out. And the models, from the model, we can give a sequence, a genomic sequence, and it tells us if this loci is actually a bit more open or a bit more closed, in the human case. And now once we have the human model, we can actually replace the human genomic sequence by a mutated sequence from patients that have fertility problems, or in our case, to look at something that is very drastic, at those monkey sequences. So we can replace one by one the human sequence and look at chromatin accessibility of all those enhancers for the 3 transcription factors. So we found about I think 32 potential cis-regulatory elements for those 3 transcription factors, and for each of them we did this in silico screen, and we find some very interesting patterns. So the great apes are on the left. These are the old world monkeys still on this continent. These are the new world monkeys that cross the Atlantic, and these are mice and dogs and so on. And we actually stumbled across this enhancer that was actually, we didn't really need the model actually to see it, because the sequence is absolutely absent in the New World monkeys and all the other primates and all the other mammals. This is an enhancer that appeared in the ancestor of us and of all the old world monkeys. And you can see that using the chromatin accessibility model, for example, the cynomolgus monkey, the macaque that I told you about, you can see that the expression level is actually the opening of the chromatin in this enhancer is much lower as compared to humans. And this is a putative enhancer of GCM1. So we can look at this enhancer, and the surprise was quite nice because, so this is the accessibility of the enhancer. It's higher in the polar trophectoderm as compared to the mural trophectoderm. It is very conserved in all the great apes and some old world monkeys, sorry. And then when you switch to the other monkeys and to mice, dogs, armadillo, there's absolutely no sequence. And when you look in the middle of this enhancer, you can see a beautiful sequence here that is a line 1 ALU SP line 1 sequence. And this is a viral sequence. So this ALU sequence, you can actually put a date on it. It's 35 million years ago. And this invaded the genome of our ancestors that were on this continent, multiplied as a transposon. We have millions of those sequences. And this elongated our genome massively, by the way. And one of those one of those sequences actually contributed to the formation of a new enhancer. At least that's the way we interpret it. If you look at within this enhancer, there are two motifs, a GATA2 motif on the left and a CEPB motif on the right. So those transcription factors are potentially binding to this enhancer to regulate the expression of GCM1. And that was a quite happy moment because GATA2 and CEPBD are actually quite high in our ranking. But we actually, when we knocked them out, we didn't see. We actually did not knock them out. Sorry, but we didn't see they were directly feeding into the adhesion migration programs. So we actually never knocked them out. But what we think that they are doing is actually to regulate GCM1 that is actually here. So it comes down to the possibility that those viruses that invaded our genome created this novel enhancer, and that potentially regulated the evolution of our mode of implantation. Of course, one out which is quite cool is that we can do the experiment. So we can knock out the enhancer, the core one, or we can knock out a bigger chunk that makes it marmoset-like, so we monkeyfy the human blastoids. And you can see that GCM1 is very nicely expressed on the polar side in the wild-type case. If you knock out the big chunk like a marmoset, absolutely no expression of GCM1. And if you knock out the core enhancer, absolutely no expression. But if you wait long enough, GCM1 is coming up. So by knocking out this enhancer, we are creating an heterochrony in vitro that is quite similar to what we see in the single-cell sequencing data of those monkeys. Of course, it's one enhancer. There's probably many that are acting, or a couple that are acting on this, but it clearly creates a delay that in regulating the expression of GCM1 that turns out to be crucial for the attachment of the human blastoids. So the way that we understand it is that 40 million years ago, and this we know for a long time, those monkeys crossed the Atlantic. And 35 million years ago, those ALU sequences cut and pasted themselves into the genome. This we know for quite some time already. And that this led to the evolution of the genome, and especially to the creation of an enhancer, and that this enhancer became maximally opened in the great apes, which probably led to an earlier regulation of GCM1 as compared to others, to previous species, which probably contributed to timing the attachments at an earlier stage and finding a different uterus, etc., etc. So the acquisition of this transposon-derived enhancer probably contributed to shifting the time of anchoring of human blastocysts and to increase the invasive capacity. So this is one thing that we are finishing now, and before entering discussion, I wanted to bring this into a little bit of a more general context. So the way that we are thinking about this specific mode of human reproduction is that in the sense of a fertility dilemma. So what we think is that in the course of evolution, our ancestors have acquired some genetic elements, sometimes from viral sequences, that enable the blastocyst to embed itself earlier and deeper inside the uterus. So this, there is no doubt, that we do this, the question is, whether this is a genetic system that is actually doing it, and some evidence that this is to some extent the case. And this enabled the development of larger fetuses with larger brains. So this is like this big hypothesis that is here in the field for decades. But somehow those genetic tools that we acquired are fragile, and we would love to now that we have this collection, we would love to look at the mutations that appeared in those patients with infertility, making human embryogenesis prone to failure. And very interestingly, if you think about it in terms of evolution, this vulnerability, it could be just the results of unpurposed evolutionary tinkering, like François Jacob would say, and just the random stuff that happens, and we have to cope with it, and as long as we reproduce, it's fine. But it could be also a trade-off. This often happens during evolution, that is forced by the co-evolution of different traits. And one hypothesis is that the bipedality actually influenced deeply the way that we reproduced, making the pregnancy quite risky. Because of the narrowing of the canal of delivery, and that this might have put a lot of evolutionary pressure on the way that we actually become pregnant and potentially implant our uterus and potentially make them fail. Because, and this is what one thing that we are thinking of, it is very possible also that this failure of human implantation is not something that is detrimental, but something that might have increased the fitness of our ancestors by limiting the number of pregnancies and making sure that the women actually stay alive and can take care of their children and so on. So it could be an adaptation that increased the fitness of our ancestors by limiting the frequency of pregnancy. So solving the fertility dilemma. So these are the things that we are, the framework that we are currently pursuing at the moment. And to finish, I'd just like to mention three little things. The first thing is that, of course, we are very interested in, this basic aspect, and also in evolution in general and those evolutionary hypotheses. But we are also interested in clinical research. And in order to do this, we spun out a company that's called Dawn Bio. It's actually led by Alok Javali, who's an ex-postdoc and is now the CSO in the lab. Very clever guy that I love to work with. And lately we have recruited Peter Greiner, who is the former vice president of Merck Fertility, who is now the CEO of the company. With about 10 people who are actually generating in silico screens in order to find potential pathways. And those pathways are tested using in-blastoid screens. And we actually found about 13 molecules until now. And those molecules go straight into the IVF clinic and are being tested. And out of those 13, now we have 7 that actually increase the quality of the blastocyst. These are naturally molecules that are found inside the uterus and that are thought to be, the receptor is expressed on the blastocyst, so these are natural things. And we are finishing a preclinical trial by combining some of those. But the happy take-home message for me is that blastoids are actually predictive of blastocysts. So this is a very happy moment for me. And that the second one is the IVF medium that is used currently in the clinic is very prone to optimization. And there are some major components that are currently missing. And so far we have discovered about 7 clinically relevant molecules to improve the in vitro development of human blastocysts. So this is the first little point. The second little point is that we always have doubts about the model that we are building. And we need to find ways to understand if what we find in those in vitro models are actually relevant to the real situation. And in order to do so, the way that we are approaching the problem is to work with the gynecologist, especially Jan Bronson lately at Warwick University, who has discovered that the endometrium is stratified. There's an upper layer and a bottom layer. This upper layer is for the anchorage of the blastocyst, and the bottom layer is for the formation of the cocoon. And you can look at the interaction between the different layers with the polar trophectoderm, and we can find the genes that are most likely mediating this interaction. And what we can do, and what we are doing now, this is unpublished, is to gather, to harvest the conditioned medium from human blastocysts that are grown in the IVF clinics. And we split this conditioned medium between the one that comes from blastocysts that we know by morphology are never going to form a baby, from the ones that are going to lead to a successful pregnancy. And then we take this conditioned medium and we put it on pieces of endometrium that are chopped out, and we see very different responses. This is on the right side here. So those different layers are actually capable to respond to the quality of the blastocyst. Yeah. Which is very interesting, coming back to our evolutionary hypothesis, because it looks as if the endometrium is capable of selecting the good from the bad blastocyst. Yeah. That would make a lot of sense in order to make sure that you invest into the right pregnancy and that you don't, overdo things. So this is my second little take-home message. The third one is that, of course, we are very interested into modeling the next steps, and we are doing this at the moment by building those chips with gels, stromal cells, and you can see the blastoids now like deeply embedding and forming those lacunae. So this is the next, definitely the next step. You can see that the lacunae are forming, meaning that the trophoblasts are very much differentiating and forming those cavities that are normally harvesting the blood from the mother in order to nourish the embryo. So we are now in the capacity of redoing the same exercise that I told you about to find the genes, the transcription factors, and the cis-regulatory elements that have evolved in order to mediate this process of trophoblast lacunae formation that occurs during the second week of pregnancy. I hope that in the next 2-3 years we're going to find more of those cis-regulatory elements. And of course, once we have those cis-regulatory elements, we can use them in massively parallel reporter assays. So those are extremely fine reporters that can be used to track the effect of any type of drug or molecule that is coming from the uterus or from the microbiome and so on. Yeah. So. Okay, we are doing a lot of ethics on the side. I'm happy to talk about it. Public information, because of course we are working with sensitive subjects, and we think that the three are actually coming together. And I'll finish by giving a big thank to the team. This is Haydar, who developed the implantation assay. Saurabh, we did the cis-regulatory element story. These are people working still with the mouse. Emil, Teresa, Giovanni, my lab manager, and an engineer was developing the chip, and some more people working on human projects here. Thank you very much.